CombiCult is a bead-based combinatorial stem cell screening technology capable of simultaneously testing many thousands of combinations of cell culture media in parallel. The purpose of this animation is to explain CombiCult's workflow, illustrating the instrumentation, timing and volumes of reagents required. The workflow comprises 4 integrated modules: cell culture, where beads are seeded with stem cells and exposed to multiple cell culture conditions; screening, where beads bearing differentiated cells are isolated; tag analysis, where the cell culture history of beads is determined; and bioinformatics, where experimental data are analyzed to derive optimal differentiation protocols. To begin an experiment, stem cells are seeded onto the surface of microscopic beads, which have been designed to provide an adherent growth substrate for embryonic stem cells, iPS cells, and adult stem cells. About 10 million stem cells grown in standard cell culture media are added to approximately 300,000 beads. The culture is incubated for 24 hours, during which time an average of 30 stem cells become attached to each bead, forming cell colonies. After seeding, the beads are split evenly into 10 samples. Each sample will be exposed to different cell culture conditions, for instance media containing different small molecules and growth factors, represented here by different colors. Unique fluorescent microsphere tags are used to spike each medium, allowing us to track the cell culture history. For each condition to be tested, the beads are resuspended in new cell culture media, and each culture is labeled with the corresponding tags. Plasticell has developed the beads and tags so that they bind irreversibly under standard cell culture conditions, without interfering with stem cell growth and differentiation. The tags are sterile and each aliquot quantitated, so that around 100 tags attach to each bead. Beads are exposed to the new media in an incubator for a given period, typically a few days, allowing the media to direct stem cell differentiation. After each round of cell culture, the beads are shuffled using a split pool process in order to expose them to combinations of media. To perform this practically, the bead suspensions are transferred to Labyrinth, a disposable U-shaped device manufactured by Plasticell designed to aid washing, pooling and splitting of beads. Rotating Labyrinth anti-clockwise simultaneously washes away the media from each of the cell cultures, while the beads are retained by a mesh. Labyrinth is then rotated back into an upright position, and the beads are backwashed into the device. The buffer is drained away and the washing steps repeated. After the final wash, the beads are resuspended and Labyrinth is rotated clockwise, transferring the beads into a pooling chamber, where they become thoroughly mixed with the help of fins designed to create turbulence in the liquid. By rotating Labyrinth back into the upright position, the mixed beads are split evenly into 10 new aliquots. Each Labyrinth chamber now contains an equal number of beads from all cell culture conditions tested in the previous split. The buffer solution is aspirated, leaving behind the beads which are resuspended in 10 new cell culture media, each of which is spiked with a unique tag. Using Labyrinth allows a split pool cycle to be performed in about an hour, compared to about 8 hours if the procedure is carried out manually. After the split pool step, beads are transferred back into cell culture flasks and placed in the incubator to undergo the next phase of stem cell differentiation. After the final split in an experiment, beads bearing terminally differentiated cells of interest are identified using a fluorescence assay, for example using a reporter gene, or in this case by staining with a fluorescently labeled antibody. In a typical screen, approximately 1 in every 1,000 beads returns a hit, and these beads must be sorted in order to determine their cell culture history. To do this, beads are transferred to an automated bead sorting instrument. The beads flow through the instrument in a stream and are interrogated by a laser to quantitate levels of fluorescence. We use an instrument that can read red and green fluorescence simultaneously, allowing sorting according to 2 parameters. Beads are sampled at a rate of approximately 20 per second, so that all hits can be sorted in less than a day, though if required, samples can be fixed and sorted over a longer period. The sensitivity of the instrument can identify positive beads containing as few as 5 cells. Beads bearing cells of interest are individually dispensed into the wells of a 96-well microtiter plate. The matrix of each bead is digested using a proprietary process, releasing intact tags for analysis. The microtiter plate containing free tags is loaded onto a high-throughput flow cytometer. The sample from each well is analyzed by the fax, which reads out the properties of the fluorescent tags. The fax data are read by our bioinformatics software called Ariadne. This identifies the tags present on each bead by comparing the data to a reference sample containing all tags. Ariadne determines the cell culture history of each bead and includes various bioinformatics tools which rapidly determine the optimal combinations of media for directed differentiation of stem cells. A further animation explaining the full power of Ariadne is available on Plasticell's website. CombiCult allows testing of many thousands of combinations of cell culture media, resulting in optimal stem cell differentiation protocols. As we have seen, few stem cells and minimal amounts of reagents are required. Moreover, the workflow uses widely available equipment and is easily performed in any standard stem cell lab.